Okay, we're back with this video starting at number 27 on the biology air practice test. This question, number 27, is about rabbits being introduced into Australia. They rapidly overpopulated because there weren't many predators for them. And then scientists introduced a rabbit-specific virus hoping to bring the population back in check. And it did drop the population down by a big chunk. But then, after several generations, the rabbit population began to increase again. So, what statement explains the new increase? This has to do with natural selection and evolution. In the population, there's always going to be some level of variation. And in this population of rabbits, there was some variation in their sensitivity to these viruses. How sick did they get from these viruses? Many of them got sick and died, and that explains the big drop in numbers. But some of them, maybe even though they get infected or are exposed, they don't end up getting sick. They end up staying healthy enough to survive. We would call that a natural immunity. So this answer, the best answer to this, is what explains the new increase. Some of the rabbits had a natural immunity to the virus. That was just some of the variation, that some of them were more immune than others. So the ones that were immune were sort of the ones that were left, and then since they were left and they were immune to the virus, they were the ones that were going to repopulate, and they're the ones that made the population start to increase again at the end. So instead of the virus killing off all the rabbits, it only killed off most of them, and then the ones that were left were going to start to repopulate that area. So they're the ones that pick the population back up again. So that's letter B. Next, this one's pretty straightforward. This is a DNA strand. It says each rung of the DNA ladder, so these are like the rungs of the ladder here. Each rung of the ladder is made up of, and we see that they're split in the middle because this is like an A and a T and a G and a C. It's made of a pair of nitrogen bases. We call them nitrogen bases because there's a lot of nitrogen atoms in them. Next up, this is a bit of a weird question. It's about genes that are connected to each other. We call them linked. It means literally that the genes are really close to each other on a chromosome. And when they're like that, when they're really close together, they get inherited at the same time, almost like they're the same gene. So usually we talk about like each gene is a coin flip, each gene is independent. That's like the law of segregation, the law of independent assortment. These genes are inherited separately, and so that you have a 50-50 shot of getting each gene. In this case, these genes are so close together on a chromosome that they sort of travel as a group. So if you get one, you tend to get the other one too. So what's happening here is normally you have two genes. One is for long pollen versus round pollen. That's the shape of the pollen itself. The purple flower color is dominant over red flower color. So we have P for purple, L for long, and both organisms they're going to cross here are heterozygous. Normally, when you have two that are heterozygous, you're going to get a 9:3 to 3:1 ratio. So 9:3 to 3:1 out of 16 possible outcomes. This would be like a dihybrid cross with like the 4x4 Punnett square. And again, you get 9 that are dominant. So that would be this one. You get 9 of each, purple and long. And then you would get 3 that show one dominant trait and the other recessive trait. And then 3 that go the other way with the opposite dominant trait. And then you would get 1 out of 16 that would show only the recessive traits that would be recessive for both traits at the same time. In this case, because they act like they're so close together, they act like one gene, we actually see it acts more like a 2x2 Punnett square, a regular Punnett square. So it says here that the dominant alleles, where does it say this at? Right here. The dominant traits are next to each other on the same chromosome for both plants. So the dominant alleles are side by side. That means the recessive alleles are side by side. So you would inherit those two as a set. So each parent could pass on those two things randomly. So because dominant is the more likely option, again, just like in a 2x2 Punnett square, the dominant one is probably going to show up 75% of the time. But again, this time it's both traits because you inherit them together. Getting one dominant trait and one recessive trait doesn't happen because they're right next to each other. So if you get one, you get both. Same thing for the recessive traits. And then the last one here is the likelihood of getting both recessive traits. And again, if it's just like a 2x2 Punnett square, if it's 75% of getting dominance, it's going to be 25% of getting recessive traits together at the same time. So 75-25, just like a 2x2 Punnett square. So that's that one. And this is a weird question. It's a little bit of a challenging question to answer, but that's how we make sense of that one. How could genetic engineering be used to produce a more successful crop in a hot, dry climate? Genetic engineering, a lot of times referring to that as a general statement, is talking about giving a gene to a different organism. So we could genetically engineer, for example, bacteria to make insulin for us. We can take the insulin gene, put it into bacteria, and they'll make insulin for us. In this case, they want to try to engineer a plant so it grows better if the climate is too hot or too dry to normally let it grow. So like it's a little bit more desert than the plant is used to. How could you help to make it grow? So the answer to this one is just like I was talking about. We take the DNA from a plant that's already used to dry climates, and we insert that gene, that segment that helps to make it useful in dry climates, and we insert that into the genome of the crop plant. So we insert the DNA from something else into our crop plant, and that those genes that we put in there hopefully are the ones that are going to help it to adapt to dry areas. This is what genetic modification is about. This is like when we talk about GMOs, this is what GMOs do. We take a gene that makes them a little bit more hardier in the dry weather, or a little bit stronger in cold weather, or resistant to bugs, or anything like that. We insert those genes that are going to help these plants become better in some way. So we take those genes, we insert them, and that's what a GMO organism really is. GMO, genetically modified. We take a gene that wasn't there, we put it in to hopefully help it develop that trait. So letter B. Next up, we're looking at a pedigree. It has several generations of an organism, and this is XXXY chromosome sex determination. That means XX is a female, XY is a male. Which pattern of inheritance would support the hypothesis that a trait is recessive and sex-linked? So we're talking about, this is one of our examples would have been hemophilia. We also talked about color blindness. Those are recessive and they're sex-linked. So what we see with that is males are much more likely to have these disorders than females because of the way males inherit sex-linked traits. Remember, females get 2 copies, so if it's recessive, a female could have a copy and be a carrier because she has another normal copy to cover that up. If a male gets that copy, he's going to have the disorder. So he only has to have 1 copy. So first of all, males are more likely to have it, and specifically males who have their mother's father who had the trait. Because what happens is, if the mother's father has it, he could pass it on to her and she could be a carrier and not know it. And then she could then pass it on to her son, and then he would have it because his dad would pass on a Y chromosome and he could not cover that up with a Y chromosome. So he would have that trait exposed. Even though mom might just be a carrier, the son, just like the grandfather, he wouldn't have any other X chromosome to cover it up with. So he would have it just like his grandfather. So again, males commonly will have these more often, but especially if their grandfather on their mother's side has it. So that's C. Next one up, this is about cheetahs. It says that cheetahs have about 99% the same genes. Most species, they share about 80% of like the same exact DNA, and cheetahs are about 99% the same, like the whole set of them. And so what does this lack of genetic diversity imply about their history? I hate these answers, to be honest with you. I don't like the answers that they offer, but the best answer here is that cheetahs have experienced reproductive isolation. Never experienced mutations is actually sounds like it would be a good answer, but I know that that's not the norm for them. Cheetahs they've had mutations. All species experience mutations in their genes. That's not the problem with cheetahs. What's actually happened is they've had what was called a bottleneck, where most of the cheetahs died, and so very few of them were left over. So they were in a very small, isolated group, and that's why. So they had to interbreed with each other a lot. So they're very inbred, and that's why their DNA matches so closely, because they were isolated in a small group. Again, this sounds logically like it would be a correct answer, but it is not. That is not what happened to them. They did not interbreed with other large cats, and they don't have a common ancestor with, well, they do have a common ancestor with other large cats, but that does not explain their very close similarity to each other. Reproductive isolation is the reason. A small group of them were left to reproduce together. That's why they match so closely. Alright, we're going to stop this video with this question.